is a crucial final step in plant tissue culture process. After growing in a sterile, controlled laboratory environment, plantlets are not immediately ready to survive outside conditions. During acclimatization, these plantlets are gradually adapted to natural environment factors such as lower humidity, higher light intensity, and non-sterile conditions. This process allows the plantlets to develop stronger roots, functional stomata, and better physiological stability. Proper acclimatization increases survival rates and ensures successful transfer of tissue culture plants from the laboratory to the greenhouse or field.
So this is the process of preparing tissue culture plantlet for acclimatization. First, choose vigorous plantlet. Look for 3 to 5 healthy leaf, wide robust root, and no contamination. These have the highest survival rate. Gently remove each plantlet. Rinse off all the agar from the root using tap water. Make sure no gel remains because agar can attract pathogens. Optionally, dip the root in a mild fungicide to prevent rot. Line a clear container with moist paper towel. Arrange the plantlet on the tissue without overcrowding. Close the lid closely to maintain high humidity while allowing slight heat exchange. Check daily to ensure the tissue stay damp and never flooded. Keep the plantlet here for 7 to 10 days as the root adapt and leaf thicken. Label the container and place it under low light at room temperature. Check daily to ensure the tissue.
Several days later.
After almost 1 week in the pre-acclimatization phase, the plantlets are ready for transfer to the nursery, the first true step into through conditions. We begin by selecting an appropriate pot size, small enough to prevent waterlogging but large enough to support initial root growth. The potting medium used is a sterilized soil mix amended with charcoal. Charcoal helps improve drainage and aeration, which is crucial for preventing root rot. Before planting, the soil is thoroughly moistened by soaking the pot in water. This ensures uniform hydration and reduces transplant shock. The plantlet is gently transferred into the pot. It is essential to avoid direct contact between charcoal and the roots to prevent chemical or physical damage. Each pot is clearly labeled for tracking growth and experimental consistency. Finally, the plant is placed under the partial shade. This reduces light stress and helps maintain high humidity around the leaves, easing the transition from in vitro to ex vitro conditions.
I will demonstrate the proper watering technique for plantlets during the acclimatization stage in the nursery. I will explain the process step by step, including the tools used and the correct method of watering to ensure healthy growth and survival of the plantlets. Before begin the watering process, the required materials are prepared, which include a water container and a spray bottle. These materials are commonly used in the nursery to support careful and controlled plantlets maintenance during acclimatization. Firstly, an appropriate amount of water is prepared to ensure the watering process can be carried out smoothly during the acclimatization stage. The water is then transferred into a spray bottle for easy handling during watering. This helps ensure the watering process can be done more consistently. The plantlets are spread gently and evenly, focusing on the leaf surface while avoiding excessive water on the soil. This method helps prevent leaf damage and reduce stress on the plantlets during acclimatization. Proper daily watering and care are essential to help plantlets adapt to ex vitro conditions, promote healthy growth, and increase their survival rate.
This is our plant after 1 week. In conclusion, acclimatization is a vital stage in tissue culture because it determines the survival and successful establishment of plantlets outside the laboratory. By gradually exposing plantlets to external environment conditions, they are able to adapt, strengthen their physiological functions, and grow independently. Without proper acclimatization, many tissue cultured plants would fail to survive after transfer. Therefore, careful management of this process ensures higher survival rates, healthier plants, and the overall success of tissue culture propagation.